Dear distinguished experts, good morning/afternoon. Our research group is pleased to present a review entitled Microbial Metabolites in Tumor Epigenetic Regulation. This work focuses on an important topic that has attracted rapidly growing attention in recent years, yet whose underlying mechanisms remain insufficiently and unsystematically integrated: how microbial metabolites participate in tumor initiation, progression, and therapeutic responses through epigenetic regulation. Accumulating evidence indicates that a wide range of microbiota-derived metabolites can profoundly influence host chromatin states, gene expression programs, and immune regulatory processes. However, the reported mechanisms are highly fragmented, and a unified conceptual framework is still lacking. Therefore, in this presentation, we will systematically review the sources and major classes of microbial metabolites, summarize the key epigenetic regulatory mechanisms they mediate, and further discuss their impact on tumor immune microenvironment and treatment responses. On this basis, we will explore their potential translational and clinical implications, aiming to establish an integrated, multi-level regulatory network of microbiota metabolism epigenetics tumor. Graphical abstract: traditional cancer research has often regarded epigenetic abnormalities as tumor cell intrinsic events. However, accumulating evidence suggests that the host microbiota and their metabolites represent a profoundly underestimated exogenous layer of epigenetic regulation. The aim of this review is merely to catalog individual mechanisms, but to address three fundamental questions: where do these metabolites originate? How exactly do they reprogram epigenetic landscapes? And can such regulatory processes be harnessed for clinical application? Highlights. This review conveys four central messages. First, microbial metabolites can directly serve as substrates or regulatory factors for epigenetic modifications, for example by modulating the activity of DACs and DNMTs. Second, these metabolites act not only on tumors but also reprogram immune cell functions, thereby influencing the efficacy of immunotherapy. Third, tumors are not merely passive recipients of regulation. Through metabolic reprogramming, they can in turn selectively shape specific microbial communities, forming dynamic feedback loops. Fourth, certain microbiota-derived vitamins and amino acids finely regulate DNA and histone methylation, directly affecting tumor suppressor genes and metabolic pathways. Figure 8 and 9. Histone modifications and ncRNAs. Beyond DNA methylation, microbial metabolites can also influence histone post-translational modifications as well as the biogenesis and function of ncRNAs. This enables them to act across multiple epigenetic layers simultaneously, by modulating transcriptional regulatory networks in an integrated manner. Figure 11. Remodeling of three-dimensional chromatin architecture. Epigenetic regulation extends beyond chemical modifications to include alterations in chromatin spatial organization. Microbial metabolism contributes to three-dimensional genome reorganization by modulating transcription factors and chromatin remodeling complexes. Figure 20. Clinical translation and future directions. Finally, we bring the focus back to the clinic. The key challenge for the future is not simply whether to supplement microbes, but whether metabolic epigenetic signatures can be leveraged to enable precise interventions and rational combination therapies. This study is summarized into the following aspects. This paper systematically summarizes the core mechanisms of microbial metabolites regulating tumor epigenetics and their bidirectional feedback loop. Microbial metabolites, e.g. SCFAs, secondary bile acids, affect tumor progression and immunotherapy response, histone acetylation, DNA methylation, and other modifications. Tumor metabolic reprogramming reshapes the microbiota, and microbial metabolites further intensify tumor epigenetic dysregulation, forming a protumorigenic self-reinforcing cycle. Current research faces challenges in causal dissection, individual difference, and technical integration. Multi-omics and AI are needed to drive clinical translation for precision oncology. Thank you for listening.